Hi, I'm Shashank, research scholar working under the supervision of Professor D.N. Singh at the Indian Institute of Technology, Bombay. I'm going to talk about the cementation of sands by biomineralization. This work was carried out during my secondment to the University of Strathclyde under the supervision of Dr. Charles Knapp and Dr. Grana L. Montesser. The secondment was funded by the European Union through Marie Curie Research Staff Exchange Program called GREAT. Biomineralization is a process by which organisms form minerals by creating the physical and chemical environment necessary for mineral formation and growth. In nature, biological activity leads to deposition of various minerals which cement the geomaterials. Mimicking this process in the laboratory would lead to various applications like cementation of soils, liquefaction, mitigation in sands, creating hydraulic barriers, and so on. Microbial metabolism influences the redox potential of the environment and leads to chemical changes causing sorption or leaching of certain elements in oxidized or reduced states. This leads to various processes such as ion exchange, adsorption onto surfaces, chelation of ions, or physical binding of two species or particles. But what is the origin of these type of biominerals? Biominerals are mainly of three types based on the process of deposition in the environment. Biologically controlled mineralization controls the nucleation, final location, the morphology, and other factors of the mineral deposition along with various other factors. Whereas biologically induced mineralization is a process in which the organism secretes metabolites in the environment which in turn react with ions or compounds to precipitate extracellular mineral phases. Thus, organisms do not exert any control over the mineral precipitation, including the morphology, structure, and crystallinity. On the other hand, minerals formed by the influence of external geo-environmental parameters on microbial metabolites and not directly dependent on microbial activity are known as biologically influenced mineralization. Among these, biomineralization by the process of biologically induced mineralization and biologically influenced mineralization is of much importance from the geoenvironmental perspective. Microbially induced calcite precipitation is a biologically induced mineralization process and is a classical example of cementation and genesis of calcareous rocks in nature. Researchers have attempted to mimic this process in a laboratory under controlled conditions with a view to employ it as one of the ground improvement and ground creation techniques. As a consequence of microbial metabolic activity, calcite precipitation occurs. Enzymatic hydrolysis of urea by microbes is the most commonly adopted techniques among various other techniques available. Precipitation of calcium carbonate by urea hydrolysis involves the production of carbonate ions in the presence of calcium ions. Calcium carbonate precipitates due to the increase in pH by the ammonium ions. Two main applications of this method that could be highlighted here with this context are the biocementation and bioclogging. In biocementation, there exist key links or connections between the adjacent soil particles which bind them together to form a porous cemented matrix. Clogging involves the filling up of the voids in the sand matrix completely by microbial calcite, which makes it an impervious sand matrix. Study concentrated mainly on some of the following aspects: optimizing several parameters such as concentration of bacteria, concentration of cementing solution, and frequency of injection into the soil matrix. Since it is the urease enzyme which is doing the trick, an alternative source of urease enzyme was considered for comparison with microbial cementation, and all this leading to understand how the cementation would affect the hydraulic conductivity of soils. From this study, it was observed that the hydraulic conductivity reduced by 3 orders of magnitude. The enzyme treatment was found to be effective as compared to microbial treatment. The depth of penetration of calcite deposition has always been an issue with respect to the uniformity in deposition. This needs to be further investigated. Thank you.